Okay, get ready. A deep dive into a story. Well, it's part scientific brilliance, part sheer luck. The discovery of penicillin. You might think you know this one, but trust me, we're going to uncover some fascinating details and really get into the how and the why. This moldy mishap changed medicine forever.
You know, it's always captivated me about this story, how it highlights the winding path of scientific discovery. It wasn't a straight line from A to B, but rather a series of unexpected turns, chance encounters, persistent problem solving, ultimately leading to a medical revolution.
So let's start at the beginning, with the man who first noticed something peculiar happening on a petri dish, Alexander Fleming. Now his journey to becoming a medical researcher.
Yeah.
Anything but typical. I mean, who knew marksmanship could be a gateway to bacteriology?
It's a fascinating example of how seemingly unrelated skills and passions can converge in unexpected ways. Fleming's sharpshooting talent caught the attention of his rifle club captain, who then connected him with a prominent researcher at St. Mary's Medical School, Sir Almroth Wright. This chance encounter led Fleming to join Wright's research and eventually become a lecturer at St. Mary.
It makes you wonder, right, how many potential breakthroughs are out there just waiting for that chance encounter or that nudge in the right direction. But Fleming's story doesn't end there. His experiences during World War I had a profound impact on his research trajectory. You know, sometimes adversity really fuels innovation.
Absolutely. Fleming served in the Royal Army Medical Corps during the war, where he witnessed firsthand the devastating impact of infected wounds. The antiseptic treatments at the time, like Flavine, weren't as effective as hoped. He even challenged their use in a 1917 paper.
So he was already questioning conventional wisdom back then. What specifically bothered him about antiseptic?
Well, Fleming observed that these antiseptics, while targeting bacteria, were also harming the body's natural defense by killing white blood. He saw how this could actually hinder the process, particularly wounds.
That's a critical observation, especially when you're trying to help the body fight off infection.
Exactly. This experience fueled his determination to find more effective to combat bacterial infections. It really laid the groundwork for his later work with penicillin.
So even amidst the chaos and tragedy of war, this seed of scientific curiosity and determination was planted. After the war, Fleming returned to St. Mary's, continued his research, focusing on antibacterial agents. He even made a significant discovery in 1922, an enzyme found in bodily fluids, like tears and saliva, that could destroy bacteria.
Ah yes, lysozyme. This discovery really highlighted the body's natural defense against infection. It was a stepping stone toward understanding how to fight bacteria more effectively.
It's incredible to think that he was already making strides in this area, years before his famous penicillin discovery. Okay, so let's get to that moment, 1928. Fleming, a bit of a messy genius, leaves some petri dishes with staphylococcus bacteria out on his lab bench before heading off on vacation.
And as fate would have it, a bit of mold decides to take up residence on one of those, forever changing the course of medical history.
That's like something out of a movie, right? Upon his return, Fleming notices something strange. The staphylococcus colonies near the mold are dissolving, while those farther away are thriving. What was going through his mind at that moment?
It was a classic eureka moment. Fleming's keen observation and scientific curiosity. He realized this wasn't just contamination. Something significant was happening.
He initially named the mold Penicillium rubrum, but it was later corrected to Penicillium rubin. Even scientific discoveries have a few rewrites along the way, right? But what's crucial is that Fleming, through meticulous experimentation, proved that this mold was producing a substance that could kill bacteria, which he named penicillin.
And what's even more remarkable is that he discovered that penicillin was incredibly potent, even when diluted. His experiments showed that even when diluted 800 times, it was still effective in killing bacteria.
Wow, that's potent. So he's witnessing this microscopic battle with the mold emerging victorious. But did he realize at that point the potential implications of his discovery?
Well, Fleming understood. He had stumbled upon something special. He even tested penicillin on himself and some lab animals, finding it remarkably non-toxic. He even tried using a crude penicillin broth to treat infected wounds and eyes in some early informal clinical trials.
So those first applications, though limited, really hinted at the therapeutic potential of penicillin. But he faced a major obstacle. He couldn't isolate penicillin in a pure form.
That's right. This stalled further progress, as he couldn't fully understand its properties or develop it into a usable drug. It's a reminder that even the most brilliant discovery often require collaboration and further research to reach their full potential.
But Fleming, being a true scientist, shared his findings with others, hoping someone could pick up where he left off. And that's where another key player enters the scene, Dr. Cecil George Payne, Fleming's former student.
Payne's work is often overlooked, but it was a critical step. He successfully used a crude penicillin broth to treat infants with eye infections in 1930, marking the first documented human use of penicillin.
Imagine that, the first human use of penicillin, with literally drops in a baby's eye. It's amazing how a former student's belief in Fleming's discovery led to this crucial step. But even with Payne's success, penicillin's full potential remained largely untapped.
That's because isolating and purifying penicillin was a formidable challenge. It would take a dedicated team with a diverse set of skills. To crack this code, and transformed penicillin from a laboratory curiosity into a life-saving drug. And that's where our next chapter begins. At Oxford University, a team of researchers led by Howard Florey, with key members Ernst Morris Chain, Norman Heatley, and Edward Abraham, took on the challenge of purifying penicillin and unlocking its true potential.
So Fleming had stumbled upon this miracle cure, but extracting it was a whole other challenge. What made this Oxford team uniquely equipped to take on this task?
Well, this team was a scientific dream team in the truest sense. Florey was a visionary leader with a keen interest in antibacterial substance chain. A brilliant biochemist, possessed the expertise to unravel the complex chemistry of penicillin. Heatley was an incredibly resourceful experimentalist, while Abraham brought his mastery of structural analysis to the table.
It sounds like they had all the right ingredients for success, so how did they finally crack the code that had eluded Fleming for so long?
Chain's ingenuity played a crucial role. He devised a method for isolating penicillin by manipulating the pH and temperature of the mold broth. Imagine coaxing this precious substance out of its hiding place through a series of precise chemical adjustments.
That sounds like a delicate and painstaking process. How much were they able to extract?
Initially, they were only able to isolate a tiny amount of purified penicillin from gallons of mold broth. We're talking fingernail-sized quantity.
Wow, that's dedication. But I'm guessing a fingernail's worth of penicillin wouldn't go very far, especially if they wanted to test its effectiveness and eventually produce it on a larger scale.
You're absolutely right. And that's where Heatley's resourcefulness really shone. He was the master of improvisation, famously using bedpans and anything else he could find. To grow more of the mold broth, this wasn't a $1 million lab. It was a team driven by the urgency of finding a solution, using whatever they could get their hands on.
So they were literally growing this miracle cure in bedpan. That's what I call thinking outside the box. But even with more mold broth, they still needed to prove that penicillin could actually save lives.
And that proof came from a rather dramatic experiment, led by Heatley. Imagine this. He spent an entire weekend meticulously monitoring a group of mice that had been injected with a lethal dose of bacteria. Half of them also received penicillin. It was a tense vigil, a true test of penicillin's life-saving power.
That must have been an incredibly tense moment in the lab. What was going through Heatley's mind during that long weekend?
He was likely a mix of nervous anticipation and cautious optimism. The fate of these mice, and perhaps the future of medicine, hung in the balance. And thankfully, the results were astounding. The mice that received penicillin survived, while those that didn't tragically succumbed to infection. It was a clear demonstration of penicillin.
I bet that moment felt electric. They knew they were on the verge of something truly groundbreaking. But as with any great scientific endeavor, the road to success isn't always smooth.
You're right. And in the case of penicillin, the story takes a somber turn with the tragic case of Albert Alexander, a policeman who developed a severe infection from a simple scratch.
His story is both heartbreaking and illuminating, isn't it? I mean, they had this potential miracle cure, but there simply wasn't enough of it to save him.
Exactly. Alexander initially showed promising improvement with penicillin treatment, but the limited supply meant they couldn't give him a full course. His death underscored the urgent need to scale up production.
It's a stark reminder that even with a breakthrough discovery, access and logistics can be major hurdles.
Absolutely. And as fate would have it, World War II became a catalyst for the mass production of penicillin. The war created an unprecedented demand for effective treatments for battlefield injuries and infections.
So necessity really drove innovation in this case. How did they manage to overcome the production challenges during wartime?
Well, it involved ingenuity, collaboration, and even a touch of desperation. Early on, they discovered that penicillin was being excreted in patients' urine in significant amounts.
So they were literally recycling penicillin from urine. Now that's what I call dedication, though maybe not the kind you'd brag about at a dinner party.
Desperate times called for creative measures, but thankfully a more sustainable solution emerged, thanks to a keen-eyed lab assistant named Mary Hunt and a moldy cantaloupe. Imagine this. She's browsing a farmer's market in Peoria, Illinois, when she spots a cantaloupe covered in what she described as a pretty golden mold.
A reminder to always keep your eyes open, even at the farmer's market. What was so special about this particular mold?
It turned out to be a strain of penicillium that produced significantly more penicillin than the strains they had been working with. Her serendipitous discovery, combined with ongoing research at the U.S. Department of Agriculture, led to a dramatic increase in penicillin production.
Talk about a lucky break. So the cantaloupe mold, coupled with the wartime push, finally allowed them to meet the massive demand for this wonder drug.
But before we move on to the impact of penicillin, I think it's important to understand how it actually works. You know, the science behind its bacteria-busting abilities.
Okay, let's dive into that. Now, you might already know about penicillin's mechanism, but let's look at it from a different angle. Imagine you're a bacterium, trying to build a sturdy wall around yourself for protection.
Okay, I'm picturing myself as a microscopic construction worker, building a fort.
Perfect. Now, penicillin comes along and acts like a saboteur, disrupting the construction of that wall. It specifically interferes with the formation of peptidoglycan, which are essential building blocks for bacterial cell walls.
So without those sturdy walls, what happens to the bacteria? They become vulnerable and essentially burst open. Penicillin targets this process, effectively killing the bacteria. And interestingly, it's more effective against gram-positive bacteria because their cell walls are more exposed.
So that's why some infections respond better to penicillin than others. It all comes down to the structure of those bacterial walls.
Precise. But as you can imagine, with the widespread use of penicillin came a new challenge: antibiotic-resistant. Fleming himself warned about this, even in the early days of discovery.
It's like an ongoing arms race, right? We develop new weapons against bacteria, and they find ways to evolve and fight back.
That's a great analogy. Bacteria are incredibly adaptable, and over time, some developed enzymes called beta-lactamase, which can break down the beta-lactam ring in penicillin, rendering it ineffective.
So even with this miracle drug, we've had to constantly innovate and develop new antibiotics to stay one step ahead of those crafty bacteria. It's a reminder that scientific progress is an ongoing journey.
Absolutely. And the story of penicillin is a testament to the power of scientific curiosity, collaboration, and persistence in the face of challenge. But it also raises important questions about responsible antibiotics and the need for continued research to combat resistance.
It's amazing to think how this one discovery, born out of a bit of messiness and a whole lot of luck, has had such a ripple effect on medicine. Speaking of recognition, the 1945 Nobel Prize in Physiology or Medicine was awarded to Fleming, Florey, and Chain for their contributions to penicillin. But some argue that not everyone who deserved credit received it.
That's right. Norman Heatley and Edward Abraham, while integral members of the Oxford, were not formally recognized by the Nobel. This highlights a common issue in science. Breakthroughs are rarely solo efforts. It often takes a village, a whole team of dedicated individuals, each bringing their unique to turn a discovery into a life-saving treatment.
Absolutely. And it's so important to acknowledge those unsung heroes, those who might not always get the spotlight, but whose contributions are just as essential to the process. But speaking of recognition, there's one tribute to Fleming that always makes me smile: the bullfighters.
Yes, you have to appreciate the irony. A group of bullfighters in Madrid, deeply grateful for penicillin's role in treating infections common to their rather dangerous profession, commissioned a bust of Alexander Fleming and a statue of a bullfighter tipping his hat in tribute.
That's a testament to the impact penicillin had across all walks of life, from soldiers on the battlefield to, well, bullfighters in the ring. It truly was a game changer.
And while Fleming, Florey, and Chain received the well-deserved Nobel Prize, we can't forget the countless others who contributed to this medical marvel: the lab assistant, the technicians, the nurses who administered the first doses. They all played a role in this remarkable story.
It really does highlight the power of collaboration and how diverse perspectives and skill sets can come together to achieve something truly extraordinary. So, as we wrap up our deep dive into the fascinating world of penicillin, what are some key takeaways you hope our listener walks away with today?
Well, penicillin's journey reminds us that scientific breakthroughs often come from unexpected places, and that even amidst setbacks and challenges, the pursuit of knowledge and a desire to alleviate suffering can lead to incredible advancements. It underscores the power of observation, persistence, and collaboration in science, and it reminds us that even with groundbreaking discoveries like penicillin, we must remain vigilant in the face of challenges like antibiotic-resistant.
It's a story of serendipity, perseverance, and the constant evolution of scientific understanding. From a forgotten petri dish to a global medical revelation, it really underscores the power of human curiosity and ingenuity.
And as we continue to explore the frontiers of medicine and face new health challenges, penicillin's legacy serves as a powerful reminder that with a bit of luck, a lot of hard work, and the spirit of collaboration, we can overcome seemingly insurmountable obstacles and improve lives for generations to come.
So as we wrap up this episode, I want to leave you with this thought. How has penicillin's story shaped our understanding of infectious disease and the development of new antibiotics? And as we face the growing challenge of antibiotic resistance, what lessons can we learn from this moldy mishap that changed the world? Keep those questions in mind as you continue your own exploration of the fascinating world of medicine and science.
It's a journey worth taking, full of twists, turns, and discoveries that have the power to shape the future of human health.
From all of us here at The Deep Dive, thanks for joining us. Until next time, keep exploring, keep questioning, and remember, sometimes the greatest discoveries come from the most unexpected places of